Come on. Ah. Alan? Emily. Ashley. John Cost. Caitlin, back in the corner. Sophia. Maria. Sam. Christina. Kristen. Sarah. Tristan. Caleb. Kyle. Jordan. Antonio. Deshaun. Jonah was there. Landon. Pat. Anthony. All right. Name of the Father, Son, and Holy Spirit, amen. God granted the Trinity to accept the things that cannot change, courage to change the things that can, wisdom to know the difference. Father, Son, and Holy Spirit, amen. All right, so move it off. Okay, real quick, let's hit these amino acids. You guys have done this. So what do you got for letter A? What do we got for letter A? Letter A. Yep, Anna. Is that the R group? What's the amino acid? What's the amino acid? We were to take a look. You guys were to go in the chart and identify all the amino acids. Did you guys do that? Okay. Did you? Yay, nay? No? Okay. All right, we're going to move on then. Moving on. You were to look up the amino acids. So let's rock and roll. Let's see here. How do I do this? Okay, those of you out there, are you seeing polymers, polysaccharides now? Yes, no?
Yep.
Okay, cool. All right, let's do this. All right, so review that. You need to be able to identify, you should be able to look those amino acids up just for practice. All right. So we got a lot to do today. Polysaccharides, more than 2. monomers going to be joined. And we are going to be mainly focusing on what's called alpha glucose, beta glucose. We talked about this yesterday, Wednesday. The bond is always going to be a glycosidic bond. Always. So the bond that holds carbohydrate monomers together is always going to be glycosidic bond. This is a glycosidic bond. This is a glycosidic bond. So we're going to name these bonds and identify these kind of bonds, first of all, based on what kind of glucose monomers are coming together to form the bond. So we notice we have alpha linkages. We're also going to see beta linkages. So you see alpha linkage, that's when alpha glucose monomers are coming together. All right, so that's the first thing. The second part of this identification tells us what carbons are being held, where that bond is being formed. So this what we're seeing here are two alpha 1,4 linkages. So what does that mean? Well, let's blow this up a little bit. Count our carbons. 1. So there's going to be a bond between carbon 1 on this glucose, alpha glucose, and 1, 4, and carbon 4 of another alpha glucose. So alpha linkages binding together two alpha glucose molecules, 1,4, that's where the bond happens. So right there, bam, there we got it. Let's do that again. We're going to add another one. Now we're going to talk about this alpha 1,4 linkage. Bam, right there. Okay, cool. Alpha 1,4. Beta 1,4. Beta 1,4, well, the beta designation tells me that this is a bond that's uniting two beta glucose molecules. Sweet. And we talked about what the difference, the structural difference between alpha and beta on Wednesday. So if that doesn't sound familiar, look at the picture, go back over and review. Beta 1,4, beta 1,4. All right? Okay. Now, the function, so that's structure. The bond is glycosidic. The functions of these polysaccharides is determined by their structure-function relationship. Alpha glucose molecules are capable of forming not just the 1,4 bond that we see here, but also this other kind of bond, this alpha 1,6 linkage. So, alpha glucose monomers are capable of forming two different kinds of alpha bonds. Alpha 1,4, alpha 1,6. So let's just take a quick look and see what an alpha 1,6 looks like. So here's our bond. So if you were asked to identify the glycosidic bond, it's the little oxygen here. All right? And I'm not even going to ask you what kind of reaction leads to this bond because you know it. All right, what is it? Dehydration. Good. By the way, every time I ask that question, That should clue you in. This is pretty important, right? I'm also trying to give you guys a little way to review and study as you're going through new things, all right? So don't just leave the old behind. All right, so we're going to form a bond between a 1 and a 6. So there's carbon 1, 2, 3, 4, 5, 6. This is carbon 6 on this alpha glucose. So that's what we're going to be forming a bond between. Yep, Deshaun. Thank you. Thank you. Awesome. Cool. So we're going to form a bond between this carbon 1 and this carbon 6 on these two glucose molecules. So I'm going to shift that over a little bit, and we would form the bond right here. Here's the oxygen that remains, and this hydroxyl and this hydrogen come off to form the can't hear that. Well, what if I take a hydrogen and I combine that with a hydroxyl, what am I going to get? Water that comes off as a result of requires the input of, thank you. See, this is easy. You just got to do it 5,000 times. Good. All right, so alpha glucose can form all kinds of different structures. It's going to give it a particular function in your cells. Molecules that are the result of alpha 1, the formation of alpha 1,4 or alpha 1,6 bonds, their function is to store energy. That's the function. Because this alpha 1,6 linkage gives the molecule the capability of forming these branched, complex carbohydrate molecules. So there's two examples, all right? We've got amylose or amylopectin. These two molecules are otherwise known as, we'd call it starch. Starch. Potatoes, you get some starch. So in plants, energy is stored as starch, amylose or amylopectin. And you and I, we store glucose as glycogen. You and I store glucose as glycogen. So starch in plants, glycogen in animals, especially us. We store glycogen in two main tissue types: skeletal muscle and the liver. So we store glycogen in two main areas: skeletal muscle and the liver. Okay, cool. Beta 1,4 glycosidic bonds. They simply form these long strands. These long strands, chains, long chains. All right? Structural support. So these are found in plants. They give plants structural support. They form long fibers, not easily soluble. Cellulose. How about celery? Celery. You bite the celery and you pull it off and it has those strings hanging off, right? You guys have done that before? That's cellulose. We call it fiber in our diet. So when you eat something from a plant source, your body doesn't digest it. It passes from one end to the next. We call that fiber. Undigestible plant material. Because we don't have the enzyme to break these beta 1,4 bonds. Okay. Cool. Questions about that? Fiber. So, carbohydrates. Oh, let's do the table. Come on, let's do the table. Come on, let's do the table. Yeah. All right. That's enough of that. Okay. So the polymer. What's the big name of the polymer? Well, we got lots of things. Polysaccharides in general, but we've got disaccharides. Actually, disaccharides. Then we've got glycogen, starch, or cellulose. Lots of different kinds of polymers here. And they all have different functions, don't they? They all have different functions. The monomers could be glucose, galactose, or fructose. Now the starch, the glycogen, the starch, and the cellulose, or no, let's just stick with glycogen. Glycogen is only glucose. All right, we don't store fructose. Okay, but monomers are glucose, galactose, and fructose. What's the bond that unites them? Glycosidic. Give me some functions. Landon. Energy storage. John Cost. Structure in plants. Good. Sarah. How about the disaccharides? What's their function? You guys remember? Helps transport that energy to get it where it needs to be. Okay. Energy can also, glucose, how about glucose? Glucose can also simply be used as energy, right? Source of energy, okay? So, good. Questions about carbohydrates? By the way, if we look at the structure here, does this thing dissolve in water or not? What do you think? Let me ask you another question. Does sugar dissolve in water? Okay, so if you can't exactly figure out from looking at this structure, think about what you actually see in real life. Oh, I put sugar in water. Wait, what do we always say? Is it raining? I don't want to go out in the rain. Why? Are you going to melt? No, we know that sugar doesn't melt, right? Are you made of sugar? Are you going to melt? Does sugar melt in water? I don't think so. What does it do in water? Dissolves in water. Why? Because all these hydroxyl and oxygen groups can form what? With water. Hydrogen bonds with water. Sweet. See, we just reviewed the last 2 weeks. All right, let's move on. Nucleic acids. To infinity and beyond. The monomer is nucleotide. You need to be able to identify all the parts of this thing. I'm telling you that. It's in red. Know it. Just know it. Like, just do it. Michael Jordan. Say, okay, I'm gonna do it. He used to stick his tongue out when he dunked. It was awesome. Anyways, so we got 3 parts. Bing, bing, bing. That's a phosphate. We know that one, right? What kind of functional group is that? What kind of functional group is that?
Phosphate.
Oh, what kind? It is a phosphate, but what kind? If you were to classify it.
In our group.
Nope. What kind of properties does it have? Is it charged, uncharged? Is it polar? It's charged.
It's charged, polar.
It's charged and it's polar. Good. Okay. This structure here is called a nitrogenous base. The purplish structure, 2 ring structures with a bunch of amino groups here. And then what's this thing down here? What's that look like? What's the blue, Anna? Sugar, da 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 da, da da da. Yeah, and we can even count it. What kind of a sugar is it? Let's count the carbons. Five. It's a pentose. So that's a sugar. Now in this little square, if it's a hydroxyl group, that makes this a ribose. But if it's lacking a hydroxyl, guess what it is? Deoxyribose. Who's heard of deoxyribose? Where do we find that? DNA, deoxyribonucleic acid. And if it's a hydroxyl, what's it in? RNA, right? Ribonucleic acid. Okay. Hey, that actually makes some sense. Okay, cool. The bond. I got to have James Bond come up here at one time. It's got to come up. Next year, it's going to come up. It's called a phosphodiester bond. So what we do is we get a bond that links the phosphate of the one nucleotide with the sugar of another. Simple. Boom. Right down there at carbon 3. So it's hooking up right there. All right. Phosphodiester bond. Function, store genetic information. That's a no-brainer. We know that one. Used for energy in most cells. Bam. ATP. You need your cells to live, boy. Da 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 da. ATP keeps me alive, right? Because it's my energy. ATP. Da 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 da. Anyone know that song? Jackson 5. Gotta love it. Okay, so remember, I'm old. All right, so that's. And then we've got, this is a weird one, transports electrons. Okay, seeing if it actually looks like that on the screen. It does. There are supposed to be bonds here. This is called NAD plus. So this is its oxidized form. We're gonna see what that means. But oxidized form means it isn't carrying electrons, so it's able to carry electrons. Transporting electrons. I'm bringing it up now. We're going to talk about its import a little bit later. But when I transport electrons, remember in a cell, the electrons, that's the particle that's what? Moving and grooving. So that's where the energy is, in a sense. So if I pick up an electron, woo, oh, I feel different. Anyways. But then if it's transporting electrons, it then hands on its electron to somebody else. It's kind of like when you hear good news. You ever hear good news? You like sort of feel better, right? And then when you hear that good news, you're like, I'm going to keep this to myself. No, you want to share it with someone else. So that way you can maybe help them feel a little better, right? So that's kind of what's going on here, right? So when you start, we're going to talk about transporting electrons. That's sort of, when a molecule receives an electron, it's getting energy, extra energy. Doesn't use it for itself, it hands it on to another molecule. Okay, cool. That was quick and easy. Sweet. So we're going to keep going. Nucleic acid is the polymer. Nucleotide is the monomer. Phosphodiester is the bond. And then we've got different functions here. Okay, different functions. Remember, these four, we're asking you to know these four for each of these molecule types. All right. Have we all taken a language at some point in our life, some sort of other foreign language, okay? So what they always do is they say, I took en français, so je vais, tu vas, il va, elle va, elle va, nous avons, nous avez, ils ont, ils vont, elles vont. And you learn like, oh, I add these endings to all of these verbs and then it's always the same. Well, they sort of suck you in. They sort of like hook you like, yeah, you know like 10 verbs now and you think you can actually like walk around and talk like, oh, and you're sort of making these sentences. And then they say, now it's time to learn the irregular verbs that don't follow any rules. That's like most of English, right? So we're going to talk about the lipids. These are weird ones, okay? So, but they have two major characteristics by which we can identify them. Lipids do not dissolve in water. Some lipids have parts of them that can interact with water, but lipids do not dissolve in water. Take some bacon fat. Add some water to your hand and try, or put it in a jar and just start spinning it or stirring it. You're not going to do it. They also have a very high number of hydrocarbon bonds, hydrogen and carbon bonds. So I see that. Why is that important? These are covalent bonds. What is their property? Covalent bond between carbon and hydrogen. It is a what kind of bond? Non-polar bond, which means it can't form what with water? Hydrogen bonds. Beautiful. So we're going to talk about a number of different kinds of lipids here today. First one are fatty acids. All right, fatty acids. So fatty acids they've got these long hydrocarbon chains. Notice on the right of both of these molecules, stearic acid and linoleic acid. Man, all we see are carbons and hydrogens. That's all we see. And then we've got this little, okay, what's this functional group on the end here? Anyone out there? What's this functional group? It's a carboxyl. That's why it's called a fatty acid, because at some point this little hydrogen can come off. Okay. And fatty acids can be either saturated or unsaturated. So who's heard of unsaturated fats? They tell you, these are good fats for you, unsaturated. They say saturated fats are bad. Well, the reason it's saturated, well, who can tell me? Well, you guys are all wearing masks, so that would end up being like a, that's exactly right. I'm joking. Actually, that's kind of what happened. So it's saturated because we notice that every bond between the carbon and its neighbor carbon are single covalent bonds. So every bond that the carbon is forming are single covalent bonds. The carbon is saturated with hydrogens. The unsaturated one, the carbons, there's at least one carbon-carbon bond that's a double bond, which means at least in some place, at least one place in this unsaturated fatty acid, the carbons are not all saturated with hydrogens. And this is going to affect the structure of the fatty acid, which is going to impact its function. That's it. So if you're being asked to identify the region that makes this an unsaturated fatty acid, you could say here or you could say here. All right. So that's a fatty acid. By the way, fatty acids, their function, energy. So what happens is that, I'm just introducing this now, so this is going to be one of those in one ear, out the other, but here's what happens. Fatty acid comes in, and it undergoes a process which is called beta oxidation. By beta, what that means is it cleaves off 2 carbon units off of the end, off of the hydrocarbon end. This would be the alpha, that's the beta, clunk, and we chop that off, and then your cell then ultimately uses this little 2 carbon unit to send through the Krebs cycle, citric acid cycle, TCA cycle, whatever you want to call that. It's got like 300 names, all right? So that's what happens to the fatty acids. Okay, so that's the first thing. So, fatty acids, here they are, saturated, unsaturated. What's their function? Used for energy. Now, you take fatty acids and you combine them to a molecule that's called glycerol. Now we're going to get something that's called a triglyceride. So, tri-3. Or if at first you don't succeed, triglyceride. No. So, tri-3 bound to a glycerol. It's easy. So, oh no. Well, we're also going to talk about phospholipids. But let's talk about triglycerides first. So this is glycerol. Ding, ding, ding. So triglycerides and phospholipids, we're going to talk about these two. They both start with glycerol. And what we're going to see is that what ends up being bound to this glycerol changes the structure of the molecule. And in this case, it really has an impact on the function that these molecules play in your body. Okay, triglyceride. Let's start with that one. 3 fatty acids bound to glycerol. Tri-3. 3-glyceride bound to glycerol. So if you see a glycerol, and what's it look like? Well, we'll get there. Now, these 3 fatty acids don't have to be the same. They can all be different. They can all be different. So what characterizes a triglyceride then is you have glycerol that has 3 fatty acids bound to it. 1, 2, 3. Boom, boom, boom. Okay? They don't have to be the same. Do I actually show up binding here? Okay. So. All right, that's Gollum. So these fatty acids, very, very much, they are hydrophobic, right? Because of all of the hydrocarbons, okay? Now, what happens is, we're gonna form bonds. We're gonna have covalent bonds forming between the glycerol in each of these fatty acids. We know that that's a dehydration reaction, so there we go. That's all I did. Everybody see that? Boom, boom. Here we go. So this is a triglyceride right here. Triglycerides, their function is to store energy. That's the function of the triglyceride. It's to store energy. In particular cells, we call them fat cells or adipose cells. Humans, our adipose cells are typically a little bit under the skin. So we get some under the skin and then around the organs for the most part. And then sometimes we store it in other places too, but primarily it's under the skin and around the organs. And as I said, the function of the free fatty acids, so this molecule here would be stored in your adipose cells, your fat cells, just waiting. And then when your body needs energy in between meals, goes out, it gets bound to it's bound to a transport protein, and ultimately these fatty acids get cleaved from the glycerol. And it's primarily these fatty acids, once they are freed from the glycerol. By the way, how do you break a covalent bond? What's that reaction called? A what reaction? A what reaction? Hydrolysis. How do you break the sticky pasta apart? Add water. Good. Okay. Ribeye steak. Yo, look at that. So these are triglycerides. And then what do you do with them? You go nom, 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 nom. You chew them up. You grind them up. Your body breaks them up. And then you can utilize those freed fatty acids. But if you don't use them right away, you store them as fat, just like the cow here, and then you break it down when you need it. All right. We're going to talk about phospholipids in a second, but let's talk a little bit about what's so special about having saturated or unsaturated fatty acids, like what the big deal is. So on the left are these On the left, is this are all of these fatty acids saturated, or do we have some unsaturated? Saturated, left, raise your hand. Use some ATP. Unsaturated. Okay, they're all saturated. We notice that representing these fatty acids as what's called a space-filling molecule, they can all line up nice and closely together. And that's really important. That gives this particular triglyceride a particular property in a body. Because hydrocarbons, what do they hate? Water. They don't hate it. They push it away. So you can imagine these three chains, these three long-ish chains, all being able to be in close proximity to one another. They can now begin to form those. What are the bondy kind of things that non-polar molecules form? Sorry, there was like a big military airplane just landed out behind you guys. Anyways, it was an old one. You missed it because you're looking at the board. So, but what are those kind of reaction-ish kind of events that happen between non-polar R groups? We call those what's? Hydrophobic interactions, right? So we got these three highly non-polar regions of these fatty acids, all able to get relatively close to one another. They can form hydrophobic interactions, and they literally can push the water out by hydrophobic exclusion. On the right, we see that these three fatty acids, I got it, they are all unsaturated. They all have at least one double bond. And what we see is that when they have got a double bond, it creates a kink in the chain. Notice the kinks. So, these three, they are hydrocarbons. If they had the opportunity, they would also undergo some pretty extreme hydrophobic exclusion interactions, but because they're so far apart from each other, they can't do it. I'm giving her all she's got, Captain. I can't form the exclusion. I can't do it. Right? So, we get reduced hydrophobic exclusion. So what? Why do I care? Why do I care? Well, lipids, or fats, lipids actually, that are comprised primarily of saturated fatty acids, they tend to be solid at room temperature. And we call them, that's a fat. So when we were looking at the steak, we saw the white fat. You can pretty much assume that when you see a piece of meat, if you see just a lump of fat, there's a lot of saturated fatty acids in that. Not a lot of water. It's pushed most of the water out. A lot of it, okay? So, if a lipid that's comprised primarily of saturated fatty acids, if they tend to be solid at room temperature, what kind of a property do you think lipids made primarily of unsaturated fatty acids will have? What do they look like at room temperature? What, water, liquid, fluid. What do we call that? What would be a liquid fat? Oil. Yeah. Boom. Okay, there's my saturated. Let's move on. Yes. An oil, olive oil, whatever. Okay? Good. Bam. We've got some unsaturated fatty acids here. Oh, how do you know, biology people? How do you know that? Because this is a lipid, and at room temperature, lipids comprised of primarily unsaturated fatty acids happen to be liquid at room temperature. Thank you very much. See, if you want to do this, you gotta go all in in geek mode. You gotta go all geek. So when you go to CAF today, see the oil, unsaturated lipids, man. See the fat, saturated lipids. You get the sugar, oh, this is probably sucrose, which means it's a disaccharide. Go all in, man. Go all in or go home. So, all right. We're gonna hit this, and then we're gonna back away for a moment because we're almost done today. All right. Phospholipids. Phospholipids. So we've done triglycerides. Well, first we started with fatty acids. Their structure. Primarily hydrocarbons. Saturated or unsaturated. All single bonds, some double bonds. Then we moved on. Triglyceride. The backbone of this molecule is a glycerol. We add 3 fatty acids to it. Its function as a triglyceride is to store energy. The function of the fatty acid that's released from a triglyceride is for energy. All right. Now, phospholipids. So notice the pink structure here, rose. What would you call that? Mauve. I don't know. I don't even know what mauve is, but I always want to say that. This is a mauve accent wall. Anyways, okay. So mauve. I'm sorry. Okay, so we've got our mauve. We have our mauve molecule, glycerol. Phospholipids only have 2 fatty acids bound to the glycerol, just 2. 1, 2. Check, check, check. 1, 2, check. 1, 2, check, check. 1, 2. I guess there's, when they're doing sound checks, if you've ever been there, it's like they only have 2 monitors. They got like a bank of like 5,000 monitors, and they're always like, check. 1, 2. 1, 2, check, check, 1, 2, whatever. Okay, so anyways. So we got 2 fatty acids, ding, ding. To the 3rd carbon in the glycerol, we've got a phosphate, and then we're gonna say this thing called a choline. Choline's got the nitrogen group up here, so pay attention to that. You definitely need to be able to draw this thing. Like the little cartoon version on the left. Fatty acid, glycerol, phosphate, choline. Know it. Know this right here. I'm telling you now. I'm telling you now. Know it, circle it, star it, be able to draw it. Okay. 2 fatty acids. And there's a phosphate up here. And then a phospholipid, the choline's also here. So what we notice then is that class is over. So we're going to pick up here. We're almost done with the molecules. So we're not done. We're not done. So for Monday, in the cell biology folder, there's a cell parts table. Does everyone see the cell parts table? Those of you who are out, you can't see this, but it's in your Schoology site in the cell biology folder. There is a link called cell parts table. Complete that over the weekend. We're going to blow through that thing on Monday. We're going to finish lipids, and then we're going to move into the cell parts. And we're going to start talking about the cell, which means that that note packet number 2, we're going to start diving into note packet number 2 on Monday. Yo! Let's keep being safe. Let's stay here. We want to stay here all semester. So thank you for the hard work. Let's keep working hard. Please help with cleaning up. Awesome.